Hello, everybody. This is Miss Peachy again from your WCA Biology B course, and we are looking at Unit 8, Lesson 3, Fossil Record and Embryology. So our key words here are comparative anatomy, comparative embryology, sedimentary rock, vertebrate embryos, vestigial structures, fossils, radiometric dating, and radioactive decay. So we're looking more, again, at different evidences that evolution has occurred and that, essentially the organisms are changing over time, right? And that we have evidence of common ancestry as well, I think. So we are talking here about comparing body structures of different organisms over time. So how can we do this? We live today, we don't live in the past. How can we compare body structures over time? We can compare body structures of living things, but using fossils allows us to compare body structures of living things to things that no longer are alive, or to look at how they have changed over time. So fossils are a great way to do this. And a fossil is the remains or imprints or traces of living things from a previous geological age. Examples include petrified bones, imprints of leaves or footprints, insects preserved in tree sap. There's lots of different kinds of fossils. Most fossils are preserved in sedimentary rock because sedimentary rock is the least damaging to any kind of remains. Essentially, an organism, once it dies, is just covered in sediment. That sediment is compacted and cemented together to kind of form a protective case, if you will, around the organism that has passed, and preserving it in a tomb of rock for as long as it would take to dig it up. When you get to different kinds of rock, like metamorphic and igneous, and you've got like a lot of heat and pressure and things that are involved that can destroy the fossil. How do we know how old it is? Well, they use a process called radiometric dating. That's where they take advantage of the fact that there are certain elements on Earth that have unstable nuclei, and those nuclei are changing. Protons and neutrons are breaking down, giving off alpha or beta particles, and that there's change that's happening, and it's occurring at a very regular rate, which is a clock essentially that allows us to determine how old something is. So we know how much of a radioactive element we would expect to see in a freshly dead organism versus how much we find in something we've dug up, and we can compare the two, and we know how fast that radioactive substance breaks down, and that gives us a clock again to be able to determine how old it actually is. And that's a very simplified explanation of radioactive decay. There are lots of fossils, and they give us lots of insights into the past. For example, the fossil of the horse is a classic example that's used in evolution. The horse has changed a lot over time. The modern horse is much both taller and has a very, it's actually, we're talking about the foot here specifically, but the teeth have changed in the horse as well. But the foot has changed remarkably as well. It has gone from a uh, primarily, with multiple fingers to just a single hoof in modern horses. And so what we're seeing here is a change in the horse leg over time. Um, and we have Orohippus, Myohippus, Hipparion, and Equus, which is our modern horse. And when we look at the graph of horse lineage, we can see each little blue dot is considered to be a common ancestor, a species. So we have I shouldn't say it's a species that's on here. Some we know of, some we don't know. We just assume they're there, but we haven't maybe found them in the fossil record yet, right? But from the evidence that we have, we can see that they all kind of branch back to this common ancestor from 60 million years ago. I'm sure I'm pronouncing it right, but Chippus. And interestingly, as an aside note, um, these animals lived in kind of an open prairie type of environment. No, excuse me. These guys lived into a forested type of environment. And so it was an advantage for them to be small so they could more easily hide from predators in the forest. They really didn't need to be that fast because they were kind of creeping around and hiding versus galloping across an open field. And that also meant that having multiple toes made it easier to kind of navigate through the uneven terrain inside of a forest undergrowth, right? As the landscape changed to more open prairie, it became more advantageous to have to move faster, so that larger middle toe became more pronounced because it gave them faster speed. And also they grew in size because longer legs meant you could run more. So eventually we have the large horse that we are familiar with today that runs very fast and is suitable for like an open prairie, not for forested undergrowth. So we can kind of see how environment plays a role in there as well. The problem is that the fossil record is not complete. It's really hard to make fossils. Fossils have to form in just the right conditions. An organism dies, it's buried very quickly after death, and it is undisturbed. And that doesn't happen most of the time. So there are probably different species, intermediate species of horses here that are predicted that we haven't found in the fossil record yet. Doesn't mean we won't ever find them, but at this point we don't have evidence of them. We actually have a pretty darn good look at the lineage of horses. It's actually one of the most complete fossil records, but there's a lot of ones we don't have as complete of a picture for. So another thing we can look at for evidence is something called vestigial structures. And a vestigial structure is something that probably had a function at some point in the past, but it no longer seems to have a function today. It's kind of neutral. It doesn't harm the organism. It doesn't help the organism. So it's not gone. It's not being selected against in any way, shape, or form. It's still present, even though it no longer performs a function. One example is the pelvic bone of whales. The whale has a small bone near the start of the tail that seems to be kind of float unattached to the rest of the skeleton. And this is a remnant of the pelvic bone when whales used to have legs to walk on land. So whales are actually supposed to have come around later after animals moved from water to land. Some animals started going back into the water, and that would have been where the whale kind of arrived from. So these vestigial structures are interesting because they show us that maybe at one point we had a use for it. Maybe we needed to have a tail in our past, which is why we have tail bones, which is the reason why maybe some people today are even still born with tails. That does happen as um, a carryover vestigial structure. It happens even in human beings. We don't have any need for tails today. They don't do us any harm. They don't do us any good. But we still have the tailbone that's present in our skeleton. Another study that scientists use is a study of comparative embryology. So we're looking at embryos of different animals. These are just little babies before they've really developed past a certain state in the egg or in utero or whatever. And what they notice is that embryos develop very similarly. The embryo of a fish develops very similarly to the embryo of a human being, which develops very similarly to the embryo of a reptile and a bird. So in the earliest stages of embryo development, it's really hard to tell the difference. If I were just to show you these pictures alone without the titles, you have a really hard time picking out which one of these embryos matched which ending organism. So when you track the cells and how they develop and the order in which things kind of come together, it's much the same in all animals, suggesting that animals have common ancestry as well, right? So again, no one piece of evidence can give us the answer, but a combination of all these different pieces of evidence really helps us to kind of put things together. And we can also look at the actual geologic time scale. So here's geochronological time scale. Wow, that's a long word here. Essentially the order in which things occurred and in which period. So we're seeing kind of a, pre-Cambrian period where we have single-celled and very simple organisms into kind of more complex organisms that are all kind of in the oceans into finally land dwellers, more complex, large reptiles, small mammals. Finally, we have kind of more of the mammal species. So we're kind of seeing a change in these organisms over time. So when we look here, they're in researchers are investigating these incredibly ancient embryos. They've actually found embryos of some of these. So fossils are kind of, they can be bones, they can be imprints, but they actually have found some preserved embryos of certain organisms that lived over 550 million years ago. So that's pretty cool. So you can take this a step further and look at ancient embryos. They've called these ancient embryonic organisms Cavacifera. I just made that. I don't know if that's how you pronounce it. I'm doing my best. And they've discovered that they develop very, very similarly to modern day embryos. They actually have the same building blocks of life that we find in modern day embryos as well. So even 550 million years ago, life developed the same. It has the same type of structure, which also tells us we must be very much related to what was alive back then as well. So that's all that this lessons kind of went through. Again, make sure you're doing the questions, make sure you're watching the little videos because I don't cover every little bit of information. But that's all I have for you all today. Have a great day, everyone, and we'll see you in the next one.